Today we're going to start the second half of our unit on energy transfer. We're going to talk about the process of cell respiration. So, photosynthesis, going back to what we've been talking about, was what plants could do to capture the energy from sunlight and trap it in the bonds of glucose. Glucose is a great molecule because glucose made in photosynthesis can then be stored as sucrose or starch or cellulose or lots of different things. It can even be converted into oils. There's all kinds of things that plants can do with glucose and that we can do with glucose, but we can store energy for days or weeks or months or years or whatever with glucose. However, and I've given this analogy before, if you think of glucose as sort of like a $20 bill, this is our glucose, it has a lot of energy packed in it, and our cell doesn't need that much energy for one reaction. So glucose is good for storing large amounts of energy, but then when your cell needs that energy, it needs to extract little bits of energy. So each little cell process, we can almost imagine each little process costing only $1. And what cell respiration is going to do is it's going to break the glucose down, break it back down into carbon dioxide and water, and extract the energy, and it's going to make ATP. And ATP is sort of like these little dollars, these little energy packets that can run a whole bunch of reactions. So we've made change for our bulky glucose molecule. Now, the formula here is, this is the overview formula for cell respiration, but you'll notice that I put a couple of little side notes here. Number one, technically, cell respiration does not have to use oxygen. It includes anaerobic respiration, which we're going to talk about in a minute, which happens without oxygen. Also, even though glucose is the sugar that we usually list as what's being used for cell respiration, technically you can run cell respiration with other carbohydrates besides glucose. You can also run it with fats, and you can also run it with proteins. But when you hear somebody say, hey, what's the formula for cell respiration, they mean this formula. For aerobic cell respiration using glucose. They don't mean, well, what if you use fat? Or what if there's no oxygen present? If they just say, what's the formula for cell respiration, they're talking about this. This is sort of the assumption. So again, cell respiration is going to break down glucose, release the energy, make ATP, and also a lot of it honestly gets released as heat. I think only about 40% of the energy from glucose is actually captured by ATP, and the rest ends up being released as heat. That's what happens in every energy transformation. Now, another misconception, keep in mind that even though this is the same as the formula for photosynthesis, except photosynthesis went the other way, keep in mind that it's not technically a reverse of photosynthesis. In other words, it's not like you take glucose and you go through the same exact steps as photosynthesis backwards. That's not what happens. The steps in cell respiration are completely different than the steps in photosynthesis, even though the overall formula is the reverse of the overall formula for photosynthesis. So I did want to comment on that. Now keep in mind that every single living thing runs cell respiration, from bacteria to fungi to animals to plants. Every living thing does some form of cell respiration. It may not be aerobic with oxygen, but every living thing does cell respiration. So it is a misconception that like plants do photosynthesis and animals do cell respiration. Not true. Plants and animals and bacteria and fungi, every living thing, every living cell does some form of cell respiration. Plants additionally do photosynthesis in the light. When light is present, they can do photosynthesis. They're still doing cell respiration at the same time. It's not like they do photosynthesis in the day and respiration at night. They do respiration 24/7, and they can do photosynthesis in the day. So another one of those misconceptions. All right, one of the main things that's going to happen in cell respiration is it's going to be a multi-step process, just like photosynthesis, and the main place where energy is going to get transferred is through these coenzymes called NAD and FAD. Now, that's not their real names. NAD, it stands for nicotine adenine dinucleotide. Again, it's one of those things where it has a really complex name, so we all just call it NAD. When NAD and FAD pick up hydrogens, they are reduced, and they now have basically energy trapped in those bonds. When they pass those hydrogens off and go back to their oxidized forms, NAD plus and FAD, they will actually, that energy will then be used to generate ATP. And you may remember that there was a coenzyme in photosynthesis too. NADP, and when it picked up hydrogen, it was NADPH. One way to remember this, and the P does not stand for photosynthesis, but sometimes you can remember that NADP is the one in plants. Think of P for plants or P for photosynthesis. And that NADH and FADH without the P, those are the ones that are in cell respiration. So NAD, when it picks up hydrogen, it temporarily holds energy. Same thing is true in photosynthesis with NADP. When it goes back to its oxidized form, it releases the energy. And this will come up later, but every NADH, when it does get rid of its hydrogen, this is going to happen at the very end of cell respiration, it'll actually generate enough energy to make 3 ATP. And every FAD will generate enough energy to make 2 ATP. And you don't need that information right now because it does come up later, but almost all the ATP made in cell respiration are going to be made by the passing off of hydrogens down an electron transport chain by NAD and FAD. All right, there are 2 ways that we can extract the energy from glucose, anaerobically and aerobically. So I wanted to quickly run through the difference. So anaerobic means without oxygen, and you'll notice right here, anaerobic does not require oxygen. Aerobic does require oxygen. Now, another thing you should know is because scientists believe early Earth didn't have any oxygen, they believe that extracting energy from glucose without oxygen actually evolved first. And it wasn't until the first plants came about, they changed the atmosphere to be the 20% oxygen that it is today, that aerobic pathways that could use oxygen evolved. The next thing you should know is the very first step in both, whether you have oxygen or not, is going to be called glycolysis. That's what we're going to be talking about tomorrow. And that particular step happens in the cytoplasm of the cell. Anaerobic respiration actually finishes in the cytoplasm of the cell. Many bacteria are anaerobic bacteria, and they actually run this kind of respiration all the time, and they never use oxygen. Aerobic respiration requires mitochondria for the last part. So aerobic uses oxygen, both start with glycolysis, but aerobic finishes in the mitochondria. So that's the difference between them. All right. Additionally, there are 2 different ways to build ATP. So we've got 2 ways to break down the glucose with and without oxygen. We've got 2 different ways to put that third phosphate on ATP. So one way is called substrate level phosphorylation. Substrate level phosphorylation, phosphorylate just means to put a phosphate on, is where an enzyme transfers phosphate from a substrate, you should recognize that word from the chapter we just did, and sticks it on a DP to make a TP. This is not very efficient. This is very, very slow. This makes ATP one at a time. But most organisms at least make some of their ATP this way. We do too. We make some of our ATP by this method. We don't make most of our ATP this way. Some bacteria make all their ATP using this method. Now, I like to give the example for this of a farmer. A farmer on a small farm could harvest his crops by hand. So he could go out there and he could pluck out everything one at a time. And that's sort of what's happening in substrate level phosphorylation. If he has a small farm, he's going to have enough time to do that. A bacteria is a small, uncomplicated little tiny thing. It doesn't need a lot of ATP. So if it makes all of its ATP by this method, which is slow, it's okay, because that's going to be enough ATP to keep the bacteria alive. Also, no oxygen is necessary to make ATP this way. So if we're in an oxygen, an environment that doesn't have oxygen, this is going to be our only option. So this is what it looks like. Literally, this is a substrate, this is an enzyme, this is the active site, you know what this is, and one at a time, phosphate gets stuck on ADP to make ATP. One at a time. Very slow, very tedious, but again, if you're a farmer on a small farm, you can pick your corn one at a time because you don't have a very big farm, so you don't need a lot of corn very quickly. But what if you're an organism that needs your stuff faster and you need a lot more? Then the ATP can be made a second way. And this second way, chemiosmotic or oxidative phosphorylation, is sort of like a farmer on a big farm. He's got machines to harvest the corn. And they can harvest lots and lots of corn at one time. Well, that's the way ATP are going to be made in chemiosmotic or oxidative phosphorylation. The name oxidative is not because of oxygen. It's called oxidative phosphorylation because it uses redox reactions. And it's called chemiosmotic because, and you may not remember this, but in the cell chapter we talked about chemiosmotic potential, or where you had a gradient, a chemiosmotic gradient, where you had a difference in charge on two sides of a membrane. So this is where this stuff is coming back, kind of combining chapters that we've done in the past. So what happens here is that NADH and FADH, technically it's FADH2, they picked up hydrogens earlier in cell respiration. They're going to get rid of all their hydrogens in chemiosmotic phosphorylation. And when they do that, those hydrogens are going to get pumped, technically just the hydrogen ions, are going to get pumped into something called the inner membrane space. Now we're actually going to come back and talk about this another day, so you don't need to know these locations right now. But this is the inner membrane of the mitochondria. And what's happening is NAD and FAD are going to get rid of their hydrogens and they're going to pump them into this. This is what they mean by the intermembrane space. It's the space between the outer membrane of the mitochondria and the inner membrane of the mitochondria. So it's all of this area in here. So the hydrogens are going to build up, and this is going to create an electrochemical gradient. And that's basically like a bunch of stored energy. The electrons, because hydrogen, technically hydrogen ion is missing its electron. The electrons are going to go through a bunch of proteins in the membrane. We'll talk more about it another time, not today. And the last thing that's going to happen is there's an enzyme called ATP synthase. And all these hydrogens that built up are going to flow back through this enzyme, back to the middle again, and that flow is going to generate ATP. And it's going to make a whole bunch of ATP at one time. So the hydrogens get pumped out, they flow back in through this enzyme, and this is an electrochemical gradient, and ATP is going to be generated. At the very end of the electron transport chain, this step, oxygen is going to pick up the hydrogens and form water. So just like how one of the first steps in photosynthesis was to break water, one of the last things, the very last thing that happens in cell respiration is you make water. The hydrogens go back to water. This is a little diagram. This is supposed to be, again, that inner membrane of the mitochondria. That's what this is. And this area right here is this intermembrane space. So NADH is going to get rid of its hydrogens. It's going to pump them out into this space. And the electrons from the hydrogens are actually going to travel down something called an electron transport chain. Once those hydrogens build up, they're going to flow back through this enzyme. This is literally an enzyme called ATP synthase. And oxygen's actually going to pick up these hydrogens and make water. And you're going to generate a whole bunch of ATP. It's actually going to make somewhere in the neighborhood of 32 ATP. The total you make in the entirety of cell respiration is about 36. So 32 of the 36 are all being made by this process. And again, this is sort of like the farmer using a machine to harvest his corn rather than picking it one at a time. It makes a lot of ATP very, very quickly. If you didn't understand this slide here, don't worry about it for now. Because when we get to this step, which is the electron transport chain, I'm going to explain it again and probably show you a little video on it and a couple of more pictures. And that's pretty much where we're going to stop today. Tomorrow we're going to talk about glycolysis and the Krebs cycle, or citric acid cycle. Those are the first two steps in cell respiration. We're going to talk about those tomorrow. And then on Friday, we're going to come back and wrap it up and talk about the electron transport chain, which is that complicated last step that I just showed you. And we'll be all done. And then your test is not until next Wednesday, so that will give us time on Monday and Tuesday to kind of summarize and put it all together.